Welcome to The Long Run. This is a podcast for biotech adventurers. I'm your host, Luke Timmerman. Today's guest is Gary Nabel. Gary is the CEO of Weston, Massachusetts-based ModeX Therapeutics. The company has a platform for making multi-specific biologics into vaccines and cancer therapies. ModeX was founded in 2020, building on work that began at Sanofi when Nabel was the chief scientific officer at that large pharmaceutical company. The initial application of this multi-specific biologic technology, currently in clinical trials, is for a vaccine against Epstein-Barr virus. This virus is known to cause infectious mononucleosis, a disease that comes with painful flu-like symptoms that can last weeks or months. But what has become even clearer in recent years is that EBV, which lays dormant in most adults, can be reactivated later in life and cause a lot of other problems. EBV is implicated as a culprit in several types of cancer, multiple sclerosis, and other autoimmune diseases. There is no vaccine against the virus, and the ModeX candidate still has a long way to go. But if ModeX and its partner, Merck, are successful, they could theoretically prevent a lot of debilitating chronic diseases. ModeX has designed other multi-specific biologics specifically to spur an immune response against certain types of cancer or to treat or prevent COVID-19. There's a fascinating story here about how deeper understanding of the biology of EBV infections and the expanding capabilities in the antibody engineering toolkit have led us to this point of possibility. Gary's career, starting as a virologist, then taking senior scientific positions at the NIH and Sanofi, has ended up leading him down this very interesting startup path. Now, before we get started, a word from the sponsor of the show, AlphaSense. So far, 2026 has been a year of transition for healthcare and life sciences. Over the first half of this year, the AlphaSense healthcare research team analyzed the individual stories moving the needle across the life sciences ecosystem as part of the Sector Spotlight series. The 8 spotlights can be downloaded at timmermanreport.com. They feature the high-stakes battles for new drug markets, like oral GLP-1s and targeted radioligand therapies, a resurgent biotech IPO market, and more. Follow the sector's key debates during the second half of 2026 and beyond with the latest sector spotlights directly on AlphaSense. And, quick question, when was the last time a CRO told you what something costs before you signed an NDA, sat through 2 scoping calls, and waited a week for a scope of work? In bioanalysis, pricing gets treated like a closely guarded secret. Dash Bio thinks that's backward. It publishes pricing publicly on its website and even has a calculator that shows how the price changes if you redesign your study. You can see what your study costs before you talk to a human being. No surprise line items, no it depends, no games. Dash was built to be the bioanalysis company that's actually straightforward to work with. Transparent pricing, guaranteed timelines, and data in days, not months. If you're tired of pricing that feels like a negotiation every single time, run your bioanalysis with Dash. Visit dash.bio/pricing and see exactly how much your next bioanalysis study will cost. Now, please enjoy this conversation with Gary Nabel on The Long Run. Gary Nabel, welcome to The Long Run.
Thank you. Pleasure to join you.
Well, Gary, I am excited to have you on this show because I love talking about infectious disease and vaccine development. And I think it could use, I think we're in an exciting moment of possibility, despite maybe what people see in the headlines. So I'm eager to learn more this hour.
That's great. That's great. It's an area that I spent a lot of time in and very dedicated to.
Okay, so let's just start with a little bit about you and how you got interested in this line of work. So from the very beginning, where are you from originally?
Well, originally from New England, from Connecticut, and I did my college in Boston and medical school in Boston as well. I would say that my interest in medical research really came during my college years. I was contemplating whether I was going to be a government major in college or a science major, biochem major. And I was actually convinced by the advisor in the government department to pursue the sciences, because what he told me was, he said, if you go into government, you'll never be able to do science. But if you go into science, you'll be able to work in government and be able to use that as a tool. And so that's what pushed me really to medical research.
Interesting. That actually came true.
That's right. That's right. I didn't believe him at the time, but it all came together.
Well, so what was it about science that got you excited from the beginning?
Well, it's interesting. When I look back, it turns out that the year I was born was the year that the structure of DNA was solved. And I think that really what happened was that we are living in what I call the golden age of biology and medicine. And I kind of grew up just being surrounded by it. And in high school, I went to a National Science Foundation program in biochemistry. I just loved uncovering the secrets of cells and how they worked, and really got hooked on really the thrill of discovery.
Who would have been a key influence, or someone maybe that inspired you as a young budding scientist?
Well, as a young scientist, to be honest with you, I was very taken with Watson and Crick. I realized that what they did fundamentally changed how we look at living organisms. And I came to know Watson later. I'm not so sure I would have been as enamored of him if I'd known his personality earlier on. But their discoveries really were the things that excited me. I would have to say, though, that if you ask me over my lifetime who the most pivotal figure for me in research was, and the mentor who made the most difference in my career, by far and no hands down, it's David Baltimore. I went to David's lab, after I finished my medical training. That's what really got me excited and interested in viruses, the interaction between viruses and the immune system. And, over the years, David became not only a mentor, but a great friend, a great constant, supporter, advisor, companion. And as with many scientists, he really was pivotal in terms of what I ended up doing.
Yeah, the late, great David Baltimore. I was privileged to know him and interview him several times, and he actually wrote a foreword for my book. But first and foremost, I mean, he was a virologist, and that led the way for him to make that discovery of reverse transcriptase, and, that altered our understanding of the central dogma, and that's what won him the Nobel Prize, and opened up all kinds of new avenues of thinking. But you decided to go down the virology and immunology route. How did that happen?
Well, I'd say the genesis of it really came when I was a postdoc in David's lab. And, at that time, HIV was erupting on the scene. And we were, aware that the HIV was selectively killing helper T cells. And I was actually both training in medicine, and yet had a window into this virus and how it worked in the lab. And my very first major paper was a paper that I published in David's lab, where we basically found the mechanism by which HIV becomes activated in T cells, and by virtue of a transcription factor, NF-kappa B. We published that in Nature back in 1987, and I'd say that really just was a stake in the ground, where again I was standing right at the intersection between the immune system and the virus, and that motivated me to just keep studying more about viruses and more about the immune system.
Okay, so this is during your postdoc time. Now, what were you thinking that you would do when you grew up? Were you thinking classical academic path? Was that the idea?
Yeah, I did an MD PhD. I was at Harvard Medical School, and then I went on from there to do a residency program at Brigham and Women's Hospital. The Brigham at the time had a program that allowed you to split your time between the lab and the clinic. So I would spend half my time in the lab, half my time taking care of patients. And, that really at the end of the day turned out to be, I think, the niche that was ideal for me. It really allowed me to bridge between people and science. And getting back to that discussion that I had with my government tutor, it was really all about having the opportunity to bring the fruits of science to benefit people. And again, vaccines, and I went on to run the Vaccine Research Center at NIH. Vaccines are really an ideal way to do that. And so I sort of viewed that I would always be trying to bridge these two worlds. And for some period of time, I actually did both of them. When I was at the University of Michigan, I was on the faculty in medicine and I saw patients. I also ran my lab. It was only after I came to NIH that I really spent all my time pursuing the science. Although when you do clinical development, particularly of vaccines and drugs, you use that clinical knowledge always. And so that balance has really been important to me throughout my career.
Did you think about like switching modes between basic and applied? Was it sort of like, switching to a different side of your brain and that they complement each other?
Yes, I did. I think that the example I like to use is, when this is pretty biological and somewhat nerdy, but, if you look at, the development of a red cell, when it decides to switch from making fetal hemoglobin to adult hemoglobin, it doesn't happen all at once in one day, right? For a while, the cell makes fetal and then it makes adult for a few days, and then it switches back and forth, and then it spends more time making adult, and eventually it just makes adult hemoglobin. For me, that's kind of the way the process worked. When I was earlier in my training, I would go back and forth, and I would be in the lab for a while, then I'd be in the clinic for a while, then I would spend a little more time in the clinic, then a little more time in the lab, then more time in the lab, and eventually, I made that shift. But I do think that, like many people, you sort of vote with your feet, and the experiences in both worlds, I think, are invaluable. And I also felt that by virtue of my training as an MD PhD student, that it was something that I could contribute to in a special way that, others who had done either just an MD or just a PhD, they could do it. But I just had a little bit more visceral experience bridging those worlds.
Now, could you say a little bit about that decision to work on vaccines? I guess that's would have started at Michigan, but then it turned into the NIH, the founding of that vaccine research center, which has gotten a lot of publicity recently. We know more about it. The public does anyway. What were you thinking, and what was the idea around that vaccine center?
Well, going back to my days with David, we realized when HIV appeared on the scene, I think these days, a lot of people forget how devastating AIDS was. I mean, the effects of the disease were brutal. People who have it, look like they have end-stage cancer, and these are people who just a few months earlier are healthy, enormously impressive physical specimens, and they're reduced, by this microscopic virus to barely being able to survive. We realized that, at the time, antiretrovirals were coming along, but they were early. And traditionally, for viral infections, the best way to contain them is by making a vaccine. So but the efforts at making a vaccine against HIV were thwarted by the biology of the virus. The virus was constantly mutating. It was, essentially a shifting target. The proteins on the virus were constantly changing. And so it really defied all of the criteria that you would normally have to at least quickly make a vaccine. So the thought leaders in the field, that included David, it included Tony Fauci, it included Harold Varmus, Bill Paul. They felt we needed a special effort to really approach this vaccine in a way we didn't approach vaccines in the past. And they decided, in fact, President Clinton announced in a speech, and I think it was 1998, that he wanted to establish a vaccine research center. And then he fast-tracked it. And then they went and looked for a director. And initially, I had no interest in becoming the director. But a number of people declined the position, and a few of them recommended me for the job. And initially, I thought, nah. We lived in Ann Arbor, Michigan. I was on the faculty. I was very happy there. My wife had a position and was very happy there. Our kids were in school there. And but when I saw what the NIH was prepared to do, understanding the magnitude of the problem, understanding the opportunities to really use our knowledge of science and the power of government to, put resources behind the problem, I'd say that was the point at which I decided, it's time to go all in on the vaccine side. And that's when I was asked if I would lead the Vaccine Research Center, and literally we built it from the ground up. It was there was no building, there was a hole in the ground, and we built the building. We recruited the investigators, and that really was the starting point of what I would call my most intense efforts in not just vaccines, but I would say immunotherapy in general. And having, trained as an immunologist originally, I think immunologically. And so that's how it all came together.
So here you came to government after all those years. You wouldn't have to write any more NIH grant proposals, but I guess you would need to make your case for the annual appropriation and continue to persuade folks that the efforts were worthwhile for developing vaccines for HIV, flu. I think or anything else that might pop up. Right, quickly you'd have to respond, whether it be SARS or Ebola or something else.
That's exactly what did happen. You're right, because when we established the center, we established it, to really promote the development of an HIV vaccine. But we also said, listen, the tools that we're using to develop an HIV vaccine can be applied equally well to any number of infectious disease threats. So quite early in the process, my lab and at the time, my postdoc, who became an independent investigator, Nancy Sullivan, and a highly respected scientist in the field, we worked on Ebola vaccines and developed some of the first prototypes. When avian flu erupted unexpectedly, we developed prototypes for avian flu, West Nile virus, chikungunya virus, Zika virus, ultimately the second SARS virus. The work on the second SARS virus came after I had left the VRC, but the ground that was prepared for that just by the general approach that you're suggesting.
Sure, all those things bring me back to the 2000s and 2010s. But now, can you say a little bit more about the tools that were becoming available in those years you were there at the VRC? Because I think people, vaccines, if you go way back into history, there was kind of two ways of making one. There was the whole killed, and then there was the live attenuated. And, they had their limitations. But what was getting better? That was giving you more confidence that you could you could make more effective vaccines more quickly.
Well, there were a couple of developments that I think were key, and there were a couple of features of HIV that really forced us to take a different approach. So, for example, if you tried to take an inactivated, even a whole cell or even a live attenuated vaccine for HIV, and believe me, it was tried and it failed miserably, you realize that the virus had evolved to outsmart the immune system. And so rather than using the old, time-tested approach of taking the virus and modifying it or somehow inactivating it, like Jonas Salk did or Albert Sabin did for poliovirus, we had to say, you can't do that here. What you have to do is you have to pursue what we called rational vaccine design. And that is, you need to understand the basic biology of the virus, the mechanism by which it infects a cell, the counter responses of the immune system. And then you had to use that to pinpoint the vulnerabilities of the virus and be very surgical about how you would now prevent the virus from gaining a foothold within a cell. And among the scientific advances, one of the big ones were the advances in structural biology. We were able for really the first time to start to see the structure of the viral glycoproteins at the atomic level, and then pinpoint some of those levels of critical vulnerability. We had Peter Kwong at the Vaccine Research Center, who was a leader in that. There were others around the country, Ian Wilson, Steve Harrison, who were, leaders in that field. But it was a major jump forward in our understanding. We also learned a lot about, how B cells respond to infection. And so, for example, the new technology in defining the antibodies that come from B cells and the rare antibodies that mediate protection, that was key to making progress. So there was really a confluence of progress, and I should obviously can't leave out one of the most other fundamental areas, which is the genetics, having the ability to sequence and see how the virus was evolving. They all became part of the toolkit. And with that and this approach to rational vaccine design, I think it enabled a lot of progress against many different pathogens.
And you had recombinant DNA too. So if you had a sequence that you knew was important to make a certain protein, you could take bits of the protein or one versus another, and you could mix and match and see what elicited the best type of immunity you were looking for.
Oh, absolutely. Recombinant DNA was key, and the advances in understanding using DNA as a vaccine subsequently, the wonderful work with using mRNA as a vaccine. They were all really essential advancements.
So the VRC, I mean, you were unable to develop an HIV vaccine as of yet. It doesn't exist. But there were plenty of other, I mean, you laid the groundwork, the people, the tools, the methods, everything was coming together there for a period of years. And we saw that pay off with COVID and that response, which I think surprised everyone in terms of its ability to move quickly with the next generation technology, mRNA. But you had left prior to this, and you went to industry to go work at Sanofi. Can you just briefly say a little bit about why you went there and what you sought to do?
Yeah, I went to Sanofi really for a couple of reasons. First of all, we did, even before I left, we did make use of these technologies to generate vaccines that have since become licensed, that even before COVID. The work on structure-based vaccine design that came from Jason McClellan, Barney Graham, Peter Kwong, that led the way to the RSV vaccine that we have today. My lab worked on a prototype chikungunya vaccine that has since been approved and is out there and available to the public. And then, as you point out, the SARS also really has made an impact. But one of the things I realized around about 2012 or so was that the problem of HIV, or at least a vaccine for HIV, was so daunting that I didn't think that it was likely we would solve it in the next 10 years. And I wanted to, before my career was over, I mentioned how I really wanted to bring the fruits of our science and have them really bring some value to patients. I felt I could best do that in industry. First of all, even as good as the Vaccine Research Center was, and it was great, and the people were phenomenal, their strength is not in scaling. And if you want to make a vaccine that can get out, or you want to make a drug that can get out and reach millions, tens of millions, hundreds of millions of people, that requires special technology, and that's really only available in industry. So I felt that was something I wanted to do. I felt I could learn and contribute. And in fact, that's what happened at Sanofi. I was the chief scientific officer. I was involved in bringing drugs forward, often some internal to Sanofi, some collaborations, for example, with Regeneron, that produced drugs that really got out there and helped people. Dupixent for asthma and allergic conditions. Cerulumab for inflammatory, an IL-6 antibody, some new forms of insulin, some vaccines. So I felt that there was less of a gap and more of an opportunity to see things across the finish line and to continue to learn. So that's what I enjoyed about that transition.
Dupixent, one of the biggest selling drugs in the world. So yeah, that's one way to measure your impact. But now so as a CSO, you've got this whole portfolio to oversee at a large pharma company. Now this would have been, I think you were there about 8 years, 2012 to 2020. You also oversaw this thing called Breakthrough Laboratories. And you were working with Elias Zerhouni, I think, on this in those years. What was the idea there with Breakthrough Laboratories?
Yeah, well, Elias was the head of NIH when I was the director of the Vaccine Research Center. And we got to know each other well, and obviously had many common philosophies and approaches to science and medicine. And he asked me he was the president of R&D. He asked me if I would come to become the chief scientific officer. And I said, I'd love to. I said, but one thing, I said, I love the laboratory, I love innovation, and I'm happy to oversee large programs, but I want to have a lab there, a small lab that can do breakthrough science. We called it the Breakthrough Lab. And in that lab, we then pursued ideas that were just a little bit off the beaten path. And one of them was this idea that you could make a kind of antibody that didn't normally exist in nature. In nature, regular antibodies recognize one target. But we realized that through molecular engineering, it should be possible to make antibodies that were adapted to recognize multiple targets at once. We call them multi-specific antibodies. The very first one we developed there was for HIV, was a tri-specific antibody. But then we also developed antibodies that were immunotherapies, and particularly had applications to treating human cancers. So we developed that as one new technology. Another technology we developed was one that I had started when I was at the NIH, which was to develop different kinds of vaccines where we could mount parts of viruses on essentially biologic scaffolds. They were nanoparticles that could self-assemble and present antigens that were multiplexed and very effective at eliciting immune responses. So we worked on those. They included vaccines for flu, but importantly, nowadays, vaccines against Epstein-Barr virus, which, again, some work I had started in NIH, but then we expanded and developed new directions at Sanofi.
So far, 2026 has been a year of transition for healthcare and life sciences. Over the first half of this year, the AlphaSense healthcare research team analyzed the individual stories moving the needle across the life sciences ecosystem as part of the Sector Spotlight series. The 8 spotlights can be downloaded at TimmermanReport.com. They feature the high-stakes battles for new drug markets, like oral GLP-1s and targeted radioligand therapies, a resurgent biotech IPO market, and more. Follow the sector's key debates during the second half of 2026 and beyond with the latest sector spotlights directly on AlphaSense. And quick question, when was the last time a CRO told you what something costs before you signed an NDA, sat through 2 scoping calls, and waited a week for a scope of work? In bioanalysis, pricing gets treated like a closely guarded secret. Dash Bio thinks that's backward. It publishes pricing publicly on its website and even has a calculator that shows how the price changes if you redesign your study. You can see what your study costs before you talk to a human being. No surprise line items, no it depends, no games. Dash was built to be the bioanalysis company that's actually straightforward to work with. Transparent pricing, guaranteed timelines, and data in days, not months. If you're tired of pricing that feels like a negotiation every single time, run your bioanalysis with Dash. Visit dash.bio/pricing and see exactly how much your next bioanalysis study will cost. This is really interesting because during those years, the 2010s, there was just a lot of progress in the field of antibody engineering with bispecifics, multispecifics, like being able to make them and then also manufacture them in pretty large volumes. So you're able to ride that wave, so to speak, but also run a little lab off in the corner that is probably not that many people are paying attention to, where you could do this kind of skunkworks projects and see what these tools could really do.
Yeah, that's right. Just as we were talking about before, the detailed knowledge. So with the advent of things like single cell cloning, B-cell cloning, and PCR rescue, it became possible to, identify and create and make single antibodies from single B cells that immensely enriched our repertoire of antibodies that we could use for various therapies. So a very logical next step was, okay, we can get all of these antibodies. Oftentimes, the challenge is not getting the antibodies, but using them in the right combination. And also, as you point out, how do you put them together so that they can have good pharmacokinetics in the body, extended half-lives? How can we make them so that they are manufacturable, which means you have to make them at high concentrations and that you have to make them highly soluble. So lots of improvements in antibody engineering, and they were all coming onto the scene at that point, and it just seemed, like a logical thing to put them together.
Well, and as you point out, there was a whole lot of reductionism going on in biology. This, people want to focus rightly on, what's the single target, what's the antigen, what's the right antibody, and that can work for some settings. But the lesson from HIV and other areas was that, actually you need a multi-pronged approach, so to speak. The antiretrovirals were effective with the cocktail of antiretrovirals. And broadly neutralizing antibodies were probably a similar kind of lesson. You needed to hit the virus from multiple angles, so to speak. And these tools were enabling you to make multi-specific antibodies.
That's exactly right. I mean, what you're trying to do is, whether it's an autoimmune disease and it's a molecule like Dupixent, where you hit two major pathways in the cell, IL-4, IL-13, there you hit two with one molecule. With viruses, the name of the game is to shut off escape. And so if a virus can mutate to evade an antibody that you've made, then you've got to anticipate what it needs to do to make that escape and prevent it from happening. And that's what you can do with combinations, but probably more effectively with actual multispecifics, where you have all of the binding targets in one.
Well, this is sort of the industry thinking, right? Like, wouldn't it be great if you had a single drug that could hit 3 targets and, you dose it once a month or once every couple months and, it suppresses the virus?
Exactly. That's the goal.
Well, okay, so let's talk about Epstein-Barr virus. First of all, for folks who don't know, what is it and why is it important?
Yeah, well, Epstein-Barr virus is probably what I would call foundational in the field of virology, because it was really the first human virus that was shown to cause human cancer. So it was identified in 1962, and I think it really, put virology on the map vis-a-vis human cancer, because up to that point, cancer was a black box. We didn't know that genes could be activated to cause them. We didn't know that viruses or certain viruses could accomplish the same thing. And EBV, was well known. It was discovered, in Africa for its role in Burkitt's lymphoma, which is an early childhood lymphoma that is a B-cell lymphoma. And it actually arises in malaria-infested regions, probably because of the stimulation of B cells by the malaria, together with the transforming genes of EBV. So it's been out there, but it really was never approached as a target because there wasn't, the tools weren't there. But I do, as you were pointing out, I think that, again, there was this resurgence of technology that kind of caused us to take a second look and see if there might be a better way to approach it.
Now, Epstein-Barr virus is endemic around the world, right? I mean, everybody gets it at some point, but then it becomes latent in most people, and it can reactivate. It can flare up later. And then what happens?
Well, I think Epstein-Barr virus can do two things. So during the chronic phase of infection, it can infect two types of cells. It can infect the B cell, and it can infect the epithelial cell. And it kind of ping-pongs back and forth and amplifies very quickly. And it's really that amplification in the oropharynx, in the oral passages, that leads to the infections that you see in mononucleosis. So acutely, it can cause a very severe infectious syndrome, sore throat fever, swollen glands, and it can even in some cases be lethal. I think often it just is quite debilitating, and it causes a lot of fatigue and time to recover from it. But it can, and in its nastiest forms, it can cause serious hospitalization and even death. Once the virus completes the active replication cycle, in most people, what happens is that it then becomes quiescent, as you just described. And so it then disappears into these different cell types in a latent form. And it can go into B cells latently. It can go into epithelial cells latently, probably can go into some types of astroglial cells in the brain. And there, what it can do, depending on the host and both the genetics of the host and the environmental insults that the host may experience, it can predispose to these secondary conditions, the most serious of which is cancer. And we see probably about 200,000 cases of EBV associated cancers every year. Many of them are lymphomas. Actually, even more of them are gastric carcinomas. And then you have things like nasopharyngeal carcinoma in China, which is very frequent, Burkitt's lymphoma that I mentioned, Hodgkin's disease. So it is a nasty virus. Once the acute symptoms have resolved and you think you're in the clear. Now, not everybody will develop it, but enough people in the population do that we felt this was really a virus that needed to be dealt with. If that wasn't... Oh, go ahead.
So you have a virus here that is really widespread, and it causes mononucleosis in a certain number of people each year. It causes quite a lot of cases of cancer. We more recently have looked at records that point to an association with Epstein-Barr virus reactivation and multiple sclerosis. So there's a pretty high likelihood that it's doing something to drive multiple sclerosis. So there's a lot. It's a bad actor that manifests, it rears its head in lots of different ways.
It absolutely does. And yes, I'm glad you mentioned multiple sclerosis, because multiple sclerosis, I think there's very strong evidence now that there's a link to latent EBV infection and other autoimmune diseases. I think that there's now increasing evidence that diseases like lupus, perhaps rheumatoid arthritis, that virus, once it gets in there and it stimulates the immune system, it can break tolerance of the immune system to normal antigens. And in breaking down that tolerance, it induces these auto reactions that lead to autoimmunity.
So this is clearly a big deal. There's quite an impetus here to develop a vaccine against Epstein-Barr virus. And it's been tried, right? I mean, GSK ran a program years ago. What happened there?
Well, you point to a really important study, one that caught my eye when I was the director of the Vaccine Research Center, because GSK took one of the viral proteins, the protein that's responsible for attachment to B cells, called GP350, and they did a vaccine trial using one of their adjuvants, and they actually found a very encouraging result. They found about a 80 to 90% reduction in the incidence of infectious mono. And that by itself was a real statement and an accomplishment. But they chose not to pursue it as a vaccine, and which was something that a number of us wondered about. So the good news was that there was at least some human proof of concept that GP350 could probably do a lot of good against combating the virus. But there was something else that probably was missing, that was needed to bring it to the point where it could be used more commonly.
So this study would have been years ago. You said back at the VRC. Now, like we were just talking about, technologies had evolved, single cell sequencing, all this kind of stuff. What have you learned since about the biology that further fleshes out the story?
Yeah, so one of the things that I think gave the GSK scientists a little bit of pause was that when they looked at the people who got their vaccine who didn't get mono, they were still able to detect small amounts of virus in those individuals. And what we've learned since then is that that there's a second cell type that they had overlooked or that they hadn't targeted, and that second cell type was the epithelial cell. And it turns out that the virus uses a different receptor to get into the epithelial cell. And so as we thought about why that vaccine might not have been ideal, we realized that you probably, again, getting back into this idea of targeting multiple antigens, you probably needed to block the entry of the virus into the epithelial cell in addition to the B cell. And so there was a different protein that mediates that effect. And that's actually what prompted us to start developing prototypes. So this paper was published in, I think, 2008. In 2012, we held a meeting at the VRC to just understand the science behind EBV. And it was really during that meeting that a number of us said, wait, there's some opportunities that may have been missed. And that epithelial cell targeting was one of them.
Okay, so by the time you're at Sanofi and you've got these antibody engineering tools and you've got your Breakthrough Laboratories team, you put together a candidate, a bispecific. Is that right?
Yeah, it's what I would call a bivalent vaccine.
Okay, and what are the targets?
Well, one was a form of the GP350 protein that I mentioned to you that targets the B cell. We stripped it down so that we presented only the part that was active in binding to the receptor so we could get a more focused immune response against it. And the second was a trimer that was that's found on the surface of the virus, composed of 3 proteins. It's called GH, meaning the heavy chain, GL, the light chain, and GP42, a protein, 42,000 molecular weight. That's a trimer. And the important thing about that trimer is that it does mediate entry into the epithelial cell. Turns out it will mediate entry also into the B cell. So what we reasoned is that if we made a vaccine that contained both this truncated form of GP350 and the GHGL GP42, that would give us a pretty good chance of blocking both pathways of replication of the virus.
Okay, and so what was your product profile in mind, that this could be given in a 1 or 2 dose course in a preventive setting, or could it also be a therapeutic vaccine?
That's a great question. I think our view, and I think it hasn't changed since the beginning, is that the best time to stop a viral infection is before it ever starts. And the targets that you would choose to do that are different from the ones that you would use to treat a chronic viral infection, a latent viral infection. So that this particular vaccine is really more to prevent disease. It's possible, but I think unlikely that it could maybe do something in people who are already infected. You would pick different viral antigens to try to target infected cells. But the one we moved forward into trials was really one to try to prevent infection entirely.
I see. And I suppose you could probably give this to adolescents, before they get infectious mono.
That's exactly right. But what I would say is that's the starting point, right? Because um, we know infectious mono occurs during adolescence. We know and can target people who are at high risk for it, and we can do the studies then to show that the virus can work in that setting, and it can prevent a lot of suffering and morbidity, medical, hardship caused by the infection. But that's step one. Step two is that we ultimately want to get this in at an early age and to everybody before they ever get infected by the virus. Because as we discussed, many people get infected with EBV early in life. And so the idea would be if we can show that the vaccine prevents acute infectious mononucleosis in adolescents and that it works, and that it's safe, we can then bring down the age at which people would get it normally. And then eventually, like we do, for example, with HPV. Well, HPV hasn't gone down to early years yet, but there are other vaccines that we do give. Measles is a great example. We want to give it as early as possible. So there's what I would call step one, and there's what I would call step two. And the ultimate goal, though, would be to get it in at a relatively early age where you could protect people against all of the downstream sequelae. It would include infectious mono, it would include cancers, it would include autoimmune diseases.
Well, I suppose as the epidemiologic data continues to mount, that'll strengthen that case for turning it into a childhood vaccine. Okay, but back to your lab. I know this was happening in Santa Fe. At some point, you decided to spin this out into a standalone company, ModeX Therapeutics. What spurred that? And I guess maybe this is related, but what convinced you and others around the table that maybe you're onto something here with the EBV vaccine?
Yeah, those are great questions. Well, we talked about the power of big pharma being able to scale, but there's a limitation in big pharma that really it defeats the notion of creativity and innovation when you're trying to develop new technologies, because big pharmas have to make things at scale and get them out to people who need them. But in doing that, it consumes so many resources that oftentimes the earlier stage products that are more creative and therefore more risky can't really be supported in the way that they need to be. So that's where biotechs come in. Elias and I, throughout our time at Sanofi, talked a lot about the EBV vaccine, about the multispecific antibodies, about how we could move them forward into the clinic. And we reached a point where we said, listen, the only way we'll ever do this is by creating a company. And so we co-founded ModeX Therapeutics. And what we did at the time is we developed some of our own technology and multi-specifics, but we also realized that there were some of the products at Sanofi that would go nowhere unless we took an active role in promoting, advancing them is really the best word to describe it. So we licensed in the technology for the EBV vaccine. We licensed in a few other products. None of them really went as far because we developed newer, better technologies. But the EBV vaccine was ready for prime time. And so early on in our discussions with potential partners, we started to discuss some opportunities with Merck. And it turns out that because of their experience with HPV, They didn't have to be sold on the idea of making a vaccine to prevent cancer. They had done it, and they had the infrastructure to do it. They knew all the problems. They knew where the skeletons were in the closet. They had phenomenal experience in manufacturing, in adjuvants. We had that creative spark. We had that knowledge of, how you can move quickly and get the scientific proof of concept. So we got to the point where we said, okay, let's do a collaboration and let's also do a licensing deal. We'll carry it up to phase 1. Merck will take it over in terms of clinical development, but we'll work together all the way through. The onus shifts at some point, but together we said we'll do much better in trying to advance this quickly. And that's what we did. So the EBV vaccine candidate is now in phase 1. I think we've enrolled close to 200 patients. So we're to that point where I think we'll be ready to make decisions about phase 2 in the not too distant future. And it's really been, for me, it's been what I call an ideal collaboration where both parties bring expertise to the table and they do something together that neither one could have done alone. And it's fun. They're smart. They're experienced. They're open-minded. I'd like to think we're the same. And so it's been a real enjoyable collaboration.
Now, what have you seen so far in your preclinical data package? What were you able to show to Merck that makes you think you've got something that could really go all the way?
Yeah, well, preclinically, we tested the vaccine in several animal models, and we tested it with different vaccines. We learned about some of the critical components that we wanted, which were the ones I mentioned to you earlier. We found that in mice and in non-human primates, that we were able to induce very high titer neutralizing antibodies that prevented entry of the virus into the 2 cell types that I mentioned. Importantly, we did a study, and when I say we, some of these studies that I did were together with my colleague from NIH, Jeff Cohen, who we joined forces with back in 2014 to do the initial studies together. And we continued working with Jeff even through my time at Sanofi and getting it to the point where we felt it had legs. And one of the studies we did was a study in humanized mice where we could essentially vaccinate. Well, actually, in that case, we had to give the antibodies from animals that were vaccinated to those mice, and then we challenged them with the actual EBV virus. And it gave, basically sterilizing immunity in that model. That's about as far as you can take it preclinically. And so based on those studies, which are all published, we thought this was really a candidate worth advancing.
And what do you need to show in clinical trials to get this thing all the way across the finish line to the FDA? But maybe you don't know yet, but what do you think it's going to take?
Oh, well, I think there are several criteria that you would have to meet. And I think first and foremost, when you start out in phase 1, it's about safety. So you want to make sure you have an acceptable safety profile. And I think that's something that is typically pretty manageable, but the details are important of how you manage it. We're testing 2 different adjuvants, so we need to figure out which one of those is a better adjuvant, particularly for people who've never been exposed to EBV in the past. And then generating sufficiently high titered immune responses. I think that we would like to see immune responses that are at least comparable or greater than the antibody responses you'd see from natural infection. And then as you move through to the point where you actually do an efficacy trial, you really do need to show in the real world that by giving, you do a controlled clinical trial, half the people get vaccine, half the people get placebo, and you got to show that you're preventing the incidence of, first of all, infectious mono in that population. But there's more you can do, which is kind of the exciting part, right? Because now that we know what some of the aberrant immune responses are that give rise to multiple sclerosis or to autoimmune diseases, we can also look at those immune responses. We can also use the genetic markers to help us identify people who might be at higher risk and see if you can blunt those responses in people. So there really are a bunch of what I would call ancillary studies that can help you chart the path forward.
There's some pretty good surrogate endpoints you can use there so that you don't have to wait 20 years to say, okay, how many people got cancer, how many people got multiple sclerosis, et cetera, et cetera.
Absolutely. Absolutely. I think that, as we were talking about, we're in a new age of vaccines. We have markers, viral markers, genetic markers, immune markers. We need to make full use of them.
Now, you mentioned earlier that the same technologies that you're working with to make a bivalent vaccine in this case, they can be applied toward cancer, eliciting an immune response. We've seen this with, I think, one of the leading candidates in the pipeline right now. That's a PD-1 and VEGF-directed bispecific. There's plenty of those in the pipeline now. But you've got a toolkit to come up with novel combinations to prevent viruses from escaping the immune system or prevent cancer from developing resistance mechanisms. Can you just say a little bit about what else you're working on that excites you guys?
Yeah, thank you for asking it. We currently have 5 products that are in phase 1 from ModeX. One of those is the EBV vaccine. Another one is a multi-specific antibody directed against 2 critical components of the SARS virus that can prevent the SARS virus from growing. And we're looking to test it to see whether it can protect people who have immune impairment. And so it's one of those antiviral approaches that we talked about earlier. But then the other 3 are really molecules that are designed to recognize proteins that are found on cancer cells and then bring to those cancer cells T cells, which, are capable of killing those tumor cells. And so in a way, you can think of these antibodies, they're actually what we call tetravalent. They recognize 4 different targets at once, 2 on the tumor cell, 2 on the T cell. So we're not only bringing the T cell in proximity to the cancer cell, we're actually delivering an activation signal that sustains that T cell to kill the tumor at the site. And so we're in phase 1 for 2 of those drugs. And then there's the last one is a more general immune stimulator where we hit the major activation pathways in T cells so that we're essentially turbocharging those T cells to become activated and respond to a variety of stimuli, virally infected, subjects, cancers, the elderly. We call it a rejuvenator of the immune system. And again, it takes advantage of our knowledge of T cell biology to try to keep those cells active and sustained in the body. So it is a pretty diverse, I would call it a diversified portfolio, so that there are different product lines that are not kind of completely built on one building block. They're kind of hedged across different indications. And we're hoping that, several of them will work.
Yeah. Well, this is quite a array of projects in your pipeline for this chapter of your career? I guess your advisor way back when, I mean, imagine he would work in government and work in science. He did that, probably didn't talk about working in a startup and developing a vaccine or cancer immunotherapy, but this is a really impactful set of projects if you're able to even get, if you even get one of them across the finish line.
You're absolutely right. I think you if one works, I would be thrilled. I think, first and foremost for us, it's really all about the patients. It's all about, what can we do for people who don't have alternatives and they're struggling with life-threatening diseases. And, I think that if we can get any one of these, we'd be delighted. If more than one, we'll work hard to make it happen. But it does take a lot of hard work and a lot of talented people. I have to say the folks we have at ModeX are remarkable. John Mascola, who was the head of the Vaccine Research Center after I left and ran it and took forward the COVID vaccine we discussed. John has joined us as the chief scientific officer. Elias is still with us every day, part of our medical discussions, scientific discussions, and a whole host of everybody at all different levels. So I feel very humbled and honored to be working with these folks.
Well, these are folks that can work on a lot of different things if they want to. And, this is a sign that, here's an area that could make a big difference for a lot of people if you're successful.
Thanks. We're trying.
Gary Nabel, thank you so much for joining me on The Long Run.
Thanks for having me. I appreciate it.
Thanks for listening to The Long Run, a production of Timmerman Report. Pedro Rosado of Headstepper Media was the sound editor. Music is from D.A. Wallach. See you next episode.